So when Darwin proposed that natural selection could occur on populations, there's an assumption that there's variability within a population, and you can see the variation within a population through physical features. However, Darwin didn't know couldn't explain exactly how that worked because he didn't know about DNA or genes and alleles and so forth. Didn't know about meiosis and all these other processes. And these are the things that we're going to cover now. So not all right now in this particular lecture. What I'm going to do is break all these topics down into a series of short steps, each one covering an aspect of that whole process, going everything through, introduction of terms and some of the basics of, meiosis, how that relates to Mendelian laws, and then eventually get into alleles and frequencies of alleles in populations, and then talk about Hardy-Weinberg equation and looking at microevolution, essentially. And this is going to take several steps and several lectures to kind of get through. And so we're going to start off here with some of the basics. Okay, so we have genetically inherited material, which Mendel didn't really know. He thought there were some particles that could be inherited from one generation to the next. Same with Darwin, didn't really know about genes or DNA or chromosomes or any of those sorts of things. But this is what we have. And so we have DNA molecules within our cells. Right. The DNA molecules contain a series of codes. The codes are coded by 4 nucleotides, are A's, T's, G's, and C's. And that's it. There's just 4. So essentially there's like 4 letters, and they're spelling out all the instructions for all the proteins, essentially for all the RNA directly, and then all the proteins that then form structures within our cells that carry out all the actions and activities within the cells, and then through their action allow our metabolism to work in one way or another, or not work in one way or another, structures to form or not form, and all these sorts of things are all based on the action of proteins. So it's kind of like a little bit of the molecular side of it. Each of those proteins are coded for by some aspect of the DNA. Sometimes we have many genes. We have this term gene, and now traditionally it was a little more simple, and now it's a little more complex. So a gene is a sequence of DNA. Okay, so it's a DNA sequence. That's always going to be the case. Now, what does it code for? Well, it specifically codes for RNA. RNA, okay. RNA is that intermediate, right? Remember, talking about the RNA world. So at one point in time, maybe the first cells only had RNA. They had no DNA and they had no proteins. It was just RNA that carried everything out because RNA could do the activities, and RNA has the same sort of makeup. It doesn't have the T's, but it has A's, U's, G's, and C's, and has all the same basic components of the DNA. But in our cells and in most living things today, the DNA is what holds the information, then it's transcribed or copied into RNA. Now that RNA has many different types, and in a cell biology class you'd go into more detail, but some of those just carry amino acids. They don't make proteins, they carry amino acids. Then others of them make little molecular machines, the ribosomes, that actually build proteins. Then others carry the information for a sequence of amino acids. It's a protein. So an older, sort of out-of-date definition of a gene is that genes code for proteins. So that's not really true, because sometimes you need multiple polypeptides just to make a single protein. And so sometimes you need multiple genes just to make one protein. In addition to that, the RNA is a lot more flexible, can do a variety of different things, and can be edited and altered. So we can actually have one gene in the DNA. We can get multiple proteins from that same gene. So one gene could code for 10 proteins that actually function because there's modification that occurs afterwards. So it's a fairly complex thing that you would get into more so in more of a molecular biology, cell biology course, or genetics course. But here we're kind of introducing the terms, and I just want to make sure you're not getting misinformation or misled, right? And so genes are always DNA. They're always coding for RNA specifically, and the RNA is used to make proteins. And saying that it codes for a protein is not always going to be true. Okay, but that's what they're used to make the proteins, which express and carry out all the activities of the cell. All right, so generally that's what's going to happen. Now, you also hear the term allele. Alleles are different versions of the same gene. When we talk about Mendelian inheritance patterns, and that's what we're going to mostly be getting into in our course here. We're not going to go further beyond that. That would be in more, again, of a genetics course or more of a more upper level molecular biology course. For our purposes, we're going to have, say, just two versions. That's all Mendel really thought that there were. There were two versions, he believed, of each gene, and that their genes, these inherited traits, had two versions of them. All right, and that's what we call alleles. And so let's say this is a cell, and it's just a cell with one chromosome, really, and that's it. Now, in addition to having just one chromosome, you can say, well, what about these two things there? Well, that's because most cells are diploid. So our cells of our body, the cells of most plants that make up most of the body of the plants, except for certain types of plants. Some of the plants like mosses, their cells that make them up are actually haploid, which we'll get into that more when we talk about plants. And fungi have very different things where the actual cells that make up the body, say, of the organism are not diploid. But say for all animals and for many different plants, they're diploid cells. And most of these other organisms also have diploid cells at some point in time. And what that means is that they have two copies of each chromosome. So if this is a chromosome, that's the same chromosome. They have two of it. Now, they're not exactly the same because they came from parents. So eventually we're going to work our way around to the production of gametes and gametogenesis, and then the fertilization process, and then looking at embryological development and how tissues come from that and fates of all those sorts of things. And we'll kind of connect it all eventually. And we'll be coming back to this other times in the future. So generally, this, say, chromosome is from the mother, and this one is from the father. So maternal and paternal chromosomes. Now, so the DNA, and then within the DNA are sequences, and say in a specific site. So let's say this is the DNA here. The DNA can then be broken into regions, right? And each of these regions will represent a gene, which then codes for some type of RNA. Okay. Now, what we're going to do is focus on, say, a specific region. Specific region, say here, that is coding for a specific gene. And that gene then codes for, in our understanding here, in our explanation, we're going to say that this gene codes also for a specific trait. And it's also something that then we can see. Now, a lot of your genes and their expression, you can't see. They're different versions of enzymes in your cells, and you can't look at one person, another person, or one animal or another animal, and know that they have one version of that enzyme or not. So but we're talking about, so then, physical traits that can be observable, so that we can score them and categorize them. And if that's what Mendel was doing when he was starting to study patterns of inheritance, and that's how we're going to explain it. But just again, so that you know, traits are not only the things that you can see, there are all sorts of things that you cannot see at all that are happening inside our cells. So if this is a gene that codes for some trait on the mother's chromosome that you received, then you have another version of that same chromosome that you received from your father, and all organisms will. Now, you may have the same version of that gene on both chromosomes, or you may have two different versions. Okay, so this one could be, we usually designate them with capital letters and lowercase letters. And now this is what we'll talk about. When we talk about different alleles, we talk about dominance and recessive. So a dominant one is going to be expressed and the recessive one will be hidden. So whatever trait that this capital A1 codes for is the one that will be seen or expressed in that cell. The one that's coded for by the lowercase a here won't be seen, it won't be expressed, it'll be hidden. Okay, that's the idea. Okay. So this is just some basic terms as we kind of get going. What happens is that your cells then when they divide, go through mitosis, okay, which is basic process of cell division that results in copies of the cells that should be exactly the same. But what we're going to get into is how the gametes are formed, which can actually have a variety of different combinations, which gets at our main topic, our overarching topic, which is genetic diversity. So how do organisms become uniquely different from one another so that selection can work on them, and then new populations can have different sets of alleles and organisms that can then change over time. So Darwin describes descent with modification. So that means the offspring then can be different from the parents, and after generation and generation, eventually there can actually be great change within populations because some force was selecting for a characteristic trait or feature that was more successful than some other feature. And then this would be explained genetically. So there are these traits coded for in the DNA, and then they will be separated very randomly into the offspring. And then the offspring that have certain traits might be selected for and survive better or have more offspring themselves, and others then don't have as many offspring or they don't survive. Okay, so that's kind of, we're going to keep going back and forth putting this together. So the chromosomes have the genes. Different chromosomes can have different versions of the genes, or the alleles. And then what will start to happen, and we're going to go through the process of meiosis here, but we're going to break it into two halves because there are two parts of meiosis. So in meiosis 1, and so again, we'll talk about what is meiosis. Meiosis is going to be the process really of gamete formation. Sort of. Again, not all organisms reproduce sexually or in the way that many animals and plants do. Right? So there are variations that we'll go over within the fungi and in some groups of plants where we have some variations to this as well. And then in a whole kingdom of life, in the protista, we don't have typically the gametes for sexual reproduction at all. Right? And so biology, all living things don't do everything the same. There's a lot of variation to it. Right? So meiosis is obviously something animals are going to do. There's some animals that reproduce asexually, as well, that aren't necessarily going to do these things in this particular way. But we're talking about broader, general sorts of things. So it's generally what's going to happen, though, in meiosis is we're going to have the total number of chromosomes, which means this diploid cell with 2 of each is going to result in cells that have fewer total chromosomes, only 1 copy of each chromosome in each cell. So they'll result in haploid cells. That's not going to quite happen yet, but we're going to kind of start the whole process. So for meiosis to occur, first thing that's going to have to happen is if we're going to divide a cell, the DNA has to be replicated. So we get DNA replication, and that doubles the DNA. And so each of these chromosomes here then has another double-stranded DNA built along it. So I don't know if you could see this, I won't be able to try to fit it on here. So it's another double-stranded DNA, is built using this one as a template, and then they're actually going to be joined together by certain proteins. That's why we usually kind of see them as like a little X. They are then referred to as, so this would be the chromosome, and these would be referred to as sister chromatids. And so people will get confused sometimes when they talk about or read about sister chromatids and chromosomes and homologous chromosomes and this sort of thing. So just keep this in mind. Now, if these 2 chromosomes have the same genes on them, then they're referred to also as homologous chromosomes. All right, that they are the same. They don't have all the exact same information. They can have variation. Okay, so they can have different alleles of the same gene, but they have the same gene. So if this gene coded for the color of a flower, this one codes for the color of a flower. Right? It's not that this one codes for the color of a flower and this one codes for how tall the plant is. That's a different gene, maybe on a different chromosome. It's not here. It's not in that particular location. It's someplace else. So these chromosomes have the same genes that code for the same sorts of things, but they could have very specific differences. They'd still be called homologous chromosomes. The sister chromatids should be exactly the same. They should be exact copies through DNA replication. Now, the only way in which they would be different is if there was some mutation in DNA replication, which we'll get to later as another mechanism of genetic diversity, that there'll be mutation in copying. So a C should pair with a G, but maybe in this process it accidentally pairs it with an A, and A and C don't go together, but it just stays there and it remains, and then now the code is actually different and they're not exactly the same, but generally they should be exactly the same as one another. However, what we would have is that, this one would be the dominant big A, this one would be the dominant big A, this one would be the recessive lowercase, and this one would be the recessive lowercase. So now we have two of each. So now we have two of the dominant alleles and we have two of the recessive alleles. Okay, and because we have two sister chromatids for each chromosome. Hopefully you can follow that, because we're starting to get now the terms are start to build up. Okay. So this is all happening in a phase of meiosis called interphase 1. And they wouldn't really look like this quite yet. I'm just have to draw it like that, because otherwise you wouldn't see it. It would just be invisible, right? But that's when the replication is occurring and so forth. So then we have a process called Prophase One, and that's where the chromosomes start to look like this. They start to condense, and the reason they condense so chromosomes and sister chromatids, same thing here. They are becoming visible. So they don't become visible so that we could see them. They become visible because they are easier to separate. If they're more organized, right? They're more compact, right? If they were not condensed, then they would be very long and stringy and be very easily tangled. Right? And so they'd be very difficult to then pull apart and separate evenly without some sort of problems. So they condense. And this is the same thing that happens in mitosis. We then get into Prometaphase 1. Now, if you have studied my mitosis, you know that there are things called microtubule organizing centers, and there are only one per cell, but then during the division process, a second one is built, so there are two of them. They create little microtubules, right, called kinetochore tubules. And generally what happens is, tubules come from one microtubule organizing center and the other microtubule organizing center, and they attach to each of the sister chromatids. So in mitosis, if this were mitosis, there would be a little stringy protein structure, right, called a kinetochore tubule that would attach to here, and then it would attach to here, and then this one would attach to here, and then there'd be another one that would attach to here, and then they would start pulling, and eventually they would all line up in the middle of the cell, right, and make metaphase. That's not what happens in Prometaphase 1 of meiosis. It only partially occurs. So what happens in Prometaphase 1 is that only a single one of the sister chromatids has a kinetochore tubule attached to it. So it's not all of them, essentially. And what's going to happen is that the tubules are going to pull, and these homologous chromosomes are going to then be lined up in the middle of the cell. So what happens is kinetochore tubules attach, to one sister chromatid, not both of them, and then they start to pull on them and line them, and then they that action, that pulling on them is what lines them up, and then creates metaphase one. So in metaphase one, what happens is that homologous chromosomes pair up like this. And if there were more than one chromosome in the cell, so let's say there was another chromosome, then they would duplicate like this, and then they would be lining up here as well. Okay, so that's the idea. So that these pink ones are homologous chromosomes. They have the same genes on them. This blue one here represents homologous chromosomes having the same genes on them. One of them is from the mother, one of them is from the father, and then we have duplicated them through DNA replication, so they're sister chromatids, so there's double the amount. Right, so we're not going to do the blue ones right now. We'll come back to an example later. They're more than one chromosome, but we're just going to have it this way for right now. So, double the DNA, line up the homologous chromosomes, and then the cell is going to divide into two cells. But what's going to happen here, so again, I'll get rid of the blue ones just so you don't get confused now, because you're going to start looking for them and say, well, what about them? And that's not what we're talking about. I just tried to use that as an example to show you there's definitely going to be more than one. I'm just simplifying it. So this one comes over here. That would have been the ones from the mother. This one goes over here. This is the ones from the father. So that means in this particular cell, we have the big A. This one we have the little a, like that. And now what will occur is a second division. And without going through all the steps, it's pretty much the same sort of thing. So they kind of like decondense a little, then they recondense, and so you go through the Prophase 2 and the Prometaphase 2 and then the Metaphase 2, and then when they actually separate, that's called anaphase. And again, you should look at mitosis specifically to go through the stages. Same things happen, but there's going to be some key differences, which I'll have a separate lecture on the very specific differences between them. Just kind of going through the basics again here, terms and the names of the processes and the things that are happening. There'll eventually be another division. Again, will these same things happen all over again. And that will result in this one will divide into two cells. This one will divide into two cells. Okay. And then that means this single chromosome will go here. This one will be over here. And then this one and this one. And you'll end up with 4 haploid cells. So again, we're going to go over a variety of very specific details, things that are going to happen here, some of the stages, and some other parts of it in other separate lectures. But it's too much to really cover all at once, so that's why I'm going to be breaking it down into a whole series of pieces. Right? This is the first piece. So the first piece, again, is introducing the terms, the basic concepts, and what happens overall. So in this particular case, now we have a cell here with a dominant allele. Here's a cell with a dominant allele. Here's a cell with a recessive allele. Here's a cell with a recessive allele. And that's pretty much it. So the gametes, and so these would typically be the gametes. And that would occur through a whole separate process of spermatogenesis if they were going to be sperm, or oogenesis if they were going to become the ova or the unfertilized egg. Other things happen. All right, so they have to then these will have to get a flagella, and then mitochondria will be in there. There's a little enzyme package, a whole variety of different things, and then also certain unique characteristics and features will occur to prepare the ova as well. And we will go into some of those again in a separate lecture. But right now we're just getting the basics. So that's going to be all we're going to do for this first one, is just the introduction of some of these terms. And now, and then we're going to start to get into specifically using this overview to start to talk about Mendel, right, and laws of segregation, laws of independent assortment, and we'll be going back and forth between what Mendel observed and tried to explain, and how that correlates with what actually happens in some of the different steps and stages of meiosis. Right? And so now you just have the basics of meiosis to use as a reference.